Hi, I'm Olivier, and I'm going to be talking about a sequence-sensitive elastic network which can capture dynamical elements necessary for human microRNA maturation. But I'm also going to focus more on the method and how it can be applied to the general prediction of biomolecular function from dynamics. And so first I'm going to talk briefly about microRNA biology, then about normal mode analysis, which is the technique we are using. Then I'm going to present the whole pipeline we're using, which we call NCOM DynaSigML. And then I'm going to present to you the results of applying this pipeline to miR-125A maturation, and miR-125A is a human microRNA. So microRNAs regulate gene expression, and so this is, here you see the canonical pathway of microRNA biogenesis. And so it starts with a microRNA gene, which is transcribed in the nucleus to a primary microRNA transcript. This is then cleaved by the Drosha DGCR8 complex to generate a precursor microRNA, which is then exported, cleaved again by Dicer, and then loaded in the Argonaute protein as part of the RISC complex, which can then regulate gene expression. But today we're going to focus on the first step, the first cleavage by the Drosha DGCR8 complex. And so it has been shown that apart from just the structure of the primary microRNA, which is recognized by this enzyme, dynamics play a role in this step. So for example, here is a motif known as the GHG mismatch, which is a non-canonical base pair here, but in the equilibrium structure, it's still base pair in the microRNA, but it tends to open more, and this is somehow recognized by the microprocessor to cleave this primary transcript. So then on to normal mode analysis. So dynamics are essential to biomolecular function. If biomolecules couldn't move, they couldn't perform their functions. And these dynamics can be happening on many different time scales. So here you have just a graph showing, very fast dynamics like bond rotation up to slow dynamics, which can involve larger collective motions. And so, of course, experimental techniques can sample most of these timescales, and molecular dynamics simulations, in theory, there's no limit to the timescales we can explore, but it gets costlier and costlier as we get to long timescales, so that's why the dotted line here. Whereas normal mode analysis is kind of the reverse, where it's not very precise for slow timescale, fast timescale motions, but it can explore large timescales. And it does so by approximating the potential as a quadratic energy well. So for example, here, if let's say you have a mutation that makes conformation B higher energy than conformation A, and in the wild type is the reverse, this would be the full conformational landscape, detailed energy landscape, and normal mode analysis would approximate it with a quadratic energy well. And so these techniques are actually kind of complementary even in their definition. So molecular dynamics is very realistic, but it takes a long time to get to long time scales, whereas normal mode analysis uses an approximate potential, but it's a fast analytical solution that at once spans all time scales, and so MD is costly and NME is cheap. So in the NCOM DynaSigML pipeline, so the NCOM model is the normal analysis model we use, and I'm going to be presenting it later. But something that we are what DynaSig stands for is dynamical signatures, which are vectors that represent fluctuation at every residue, and it's a computation that we can do from all the normal modes. So what we do is we start with an input 3D structure. Here you see a 3D prediction for miR-125A. And then we apply coarse graining. So for RNA in NCOM, we have 3 masses per nucleotide. And in general, for proteins, there is 1 mass per alpha carbon. And then from the potential of the model, we solve the normal modes. So if we have n masses, we have 3n minus 6 non-trivial normal modes. And from all of these normal modes, which represent a huge linear space, we can compute dynamical signatures, which are simply fluctuation at every position. So here you see the dynamical signature mapped back on the structure, and here you see it in graph form with 3 masses per nucleotide. And so the ENCOM model is called Elastic Network Contact Model, and it's the first and only sequence-sensitive, at least to our knowledge, coarse-grained normal mode analysis model. So it has 4 terms to its potential energy, and these resemble all atom terms, but it's actually through sequence connectivity through sequential amino acids. So for example, the bond stretching term is the bond stretching of 2 sequential C-alpha carbons in the case of a protein. Then there's angle bending, dihedrals, and the long-range potential. And it's in this long-range potential that the sequence sensitivity comes from. It's actually from this beta IJ term, which is the interaction between complementary atom types, and it's at the all atom level, and it gets computed from the surface area in contact between atoms. So for example, in the case of RNA, we have 3 masses per nucleotide, but the all atom context is still taken in account, so the H bonds can be recapitulated, and the stacking also. So for example, in MIR125A, there is this G22U mutation that is a SNP in the population. And if we model it, and so we model the dynamical signatures for both the wild type and the G22U, so we don't really see on this graph, but the dotted line is the wild type, which is slightly more rigid. But if we look at the fluctuation difference, we see that the mutation makes the structure much more flexible around the mutation and around the nucleotides that are base pair in this region, so this red region here. But it also makes distal parts more flexible, like the loop region here. So the effect a mutation has can really be both local and global. And so the ENCOM dynasty ML pipeline can be used to, when mutagenesis data is available, it can be used to capture dynamics function relationships. So we start with a 3D structure for the wild type sequence, and we have to have experimental measurements of some function for sequence variance, so mutagenesis data. Then we mutate in silico. So we build in silico a 3D model for every of the sequence variants. Then for each of these sequence variants, we compute a dynamical signature, which is basically a vector. Then we feed this vector in a machine learning model which takes n inputs, and we train the model to predict the function. And so for MIR125A, the data we are using is data from the David Bartel group using a purified microprocessor. They measured the efficiency of over 29,000 MIR125A variants. And so what they did is that they exhaustively mutated each of these 6 nucleotide boxes. But mutations were never combined between 2 different boxes. And they measured the efficiency by barcoding and incubating everything together and then counting the reads to determine which variants were getting processed more efficiently by the microprocessor. And they came up with simple rules that favor processing in their in vitro system. So they saw this motif, which was already known, the mismatched GHG motif, some sequence motifs, but apart from that, they maintain that there are not really other motifs there. But our group has shown in the past that, for example, MIR125A 2D structural dynamics, they correlate really well with maturation efficiency when looking at the position of this SNP, this G22U SNP, I was telling you about. So the SNP does not change the minimum free energy structure, as you can see here, but it introduces some dynamical effects that lead to poorer maturation. So that's why we chose to study this in 3D with the DynaSigML, Encom DynaSigML pipeline, and it's really fast to do so. So for the 29,000 mutants, computing each dynamical signature takes around 3 seconds CPU. So for example, on a modern quad-core laptop, this is 6 hours CPU time. And so here, for example, you see the standardized dynamical signature value, so centered around 0, and the maturation efficiency is color-coded, and so we can see, for example, some patterns that things that are more flexible on average seem to process better. And for example, this is just zooming in on these positions. You can see the standardized values across all the 29,000 variants. And the machine learning model we're using today is a lasso regression. So it's just simple linear regression that also selects important feature, because instead of just minimizing the squared error, we also minimize the sum of the coefficients with this lambda parameter, which is regularization strength. And the advantage of this is that we can then pinpoint important features back to the structure. And so we use a hard benchmark for this to ensure the model is learning beyond the sequence. So because boxes of 6 nucleotides are mutated at once, there is a lot of sequence redundancy in the dataset. So each base pair there in red is included in this testing set, and each mutation that affects only the base pairs in blue is included in the training set. So this gives us this hard benchmark of this size. And the dynamical signatures and the MC-fold, which is 2D structure prediction software, enthalpy of folding, they are highly complementary on that hard benchmark. So alone they get R squared around 0.1, and together they get slightly above 0.2. So this it still captures something on this hard benchmark. The performance might be modest, but when we do a standard 80-20 train test split of the data, then the performance is really high, highlighting that dynamics are important, and that's what the hard benchmark is capturing. But there's also maybe some sequence patterns there that the model can capture in addition to dynamics, since it's sensitive to sequence. And if we use a simple neural network, just a multi-layer perceptron with 2 hidden layers, then it slightly outperforms the lasso model. So it gets up to close to 0.3 R squared on the hard benchmark. And we see that, for example, the enrichment of high values is better here. So the nice thing about lasso regression is that, like I said, we can map back the coefficients on the structure. So there are 3 masses per nucleotide, so phosphate, sugar, base. And here you see the coefficients from the dynamical signature, and you see the energy coefficient for the MC-fold energy of folding. So this negative coefficient means that something that is more stably folded is favored, so we'll have a higher maturation efficiency. But interestingly, when we sum the dynamical signature coefficients, they are positive on average. So it means that something that is both stable and flexible, so kind of low enthalpy but high entropy, is the most favored thing. And also the most prominent feature on the backbone is this C here, that actually corresponds to the mismatched GHG motif. So it's here in the graph. So being more flexible at this position is learned by the model as being favorable, and confirms what we already knew about this motif. So then what we can do with this model is predict the maturation efficiency of theoretical sequences. So using randomly generated variants that can combine mutations everywhere in the structure, instead of just by boxes, we predicted the maturation efficiency of 30 million random miR125A sequence variants. And so here we bin them into high folding energy medium and low folding energy. And so we visualize both the experimental data from the Bartel dataset and the predicted data from these 30 million, so 10 million for each category. And so here we plotted the maximum efficiency that was measured, because since there are so many sequences, we cannot see the bins in the histograms. And so interestingly, for the low folding energy, so the highest maturation efficiencies, we have a predicted efficiency that is higher than all that were measured in the dataset. So it could be interesting to test this experimentally, and we're actually testing some predicted sequence variants. And another important thing is that both the folding delta G and the dynamical signature are needed to explain the whole variation in maturation efficiency. So for example, here you have the same binning, and then you have the maturation efficiency as a function of folding energy. So this is the experimental data, so we can see a trend, but at the same time, the highest maturation efficiency was measured for a folding energy that is not that low. And it's this point here. So, and we can see that there is high variability in the data. And the same is true for the predicted maturation efficiencies, but of course it's a linear model, so we see this linear tendency in energy, but there is still a high variability that is due to the dynamical signature effect. And so sequence variants were predicted, which predict the maturation, the mature sequence of miR-125A, so we can then easily measure them in the lab. And our top candidate has a maturation efficiency of 2.56, so higher than anything that was measured experimentally. The folding energy is really low, minus 105. And we also have negative candidates, which have dynamical signatures that are really unfavorable, because even with folding energy lower than the wild type, the predicted maturation efficiency is lower than 0. And if we don't restrict to keeping the mature sequence, we can get really high predicted maturation efficiency, so higher than 3 with a very low folding energy. So in conclusion, 3D structural dynamics are part of the signal that is recognized by the microprocessor enzyme for primary microRNA cleavage as the first step of their maturation. Approximately 3 seconds of CPU time per sequence is needed for the application of our model, so we can test millions of theoretical sequences in a day with our Compute Canada resources. And so the next steps is to test prediction at both extremes in the lab, and we also want to generalize to other microRNAs. For example, there are data about miR-16 and miR-30 that are also available. So our NCOM DynaSigML pipeline, it can be applied to pretty much any biomolecule, so protein, RNA, protein ligand complexes, to capture dynamics function relationships. And so you can follow these QR codes if you're interested. The DynaSigML package is extensively documented online and has a tutorial. The NCOM model is ran as part of the NRG10 package, which is also extensively documented. And we have a preprint out about miR-125A maturation if you're interested. So thank you for your time. I want to thank my lab members, and especially Natalia Teruel and Gabriel Galdino, who are also presenting here. And if you found this talk interesting, I suggest you go check out their posters and their presentations. Thank you.